Hello everyone and welcome to another lecture video here in General Biology 1. Today I'll be discussing to you the cell transport. So in this particular video I'll be discussing the diffusion and osmosis. So let us first reevaluate the different parts of the plasma membrane, which is the primary structure of the cell which is responsible for the cell transport. So dito meron tayong mga proteins, the phospholipids, and the cholesterol. There is a slight difference when it comes to the classification of proteins in the context of cell transport. Before, doon sa structure ng proteins, we classify proteins in terms of their placement or location within the plasma membrane. Right now, we will classify them in terms of their function. So meron tayong marker molecules which are also known as antibodies. So sila yung tumutulong sa pag-identify ng mga molecules na gustong mag-move inside and outside of the cell. And then you have the attachment proteins, which are responsible for cell-to-cell communication and at the same time structural stability of the cells and the tissue as a whole. And then you have there the transport proteins, receptor proteins, or yung transport proteins, they are also known as carrier proteins. Yung receptor proteins naman is they are responsible for recognition. Yung marker molecules, they are responsible for giving identity to the molecule. Pero yung receptor proteins, they are responsible naman for identifying the identity. So, yung marker bigay ng pagkakakilanlan at yung receptor proteins naman sila yung kumikilala sa molecule. Yes, so they will be the one responsible for regulating the identity of the molecules. No? Kung magiging magiging compatible with the receptor, then pwede siya maka-move, no, inside and outside of the cell. So pag ma-recognize, pero pag hindi, so hindi pwede, no, i-repel ng plasma membrane. And then we have also the enzymes which makes they are the molecules, or they are the proteins that are responsible for making the chemical reaction, the rate of chemical reaction na maging mas mabilis. Okay? So, nagiging mas mabilis yung chemical reaction dahil sa mga enzymes. Yan. And then, of course, we have here the four major types of molecular movements, and ionic movements inside and outside of the cell, or yung mga methods of transportation ng molecules sa cell. So we have here diffusion, osmosis, mediated transport, vesicular transport, at saka yung other sub-methods or mga iba pang maliliit na methods under mediated transport, for example, and vesicular transport. Pero dito sa video, pag-usapan na natin yung diffusion and osmosis. So first, let's know diffusion. So ito yung movement of molecules from area of higher concentration going to area of lower concentration. So paki-take note yan, guys. The goal of diffusion is to achieve equilibrium. Ito yung kanilang goal, no? So at the end of diffusion, dapat ma-achieve yung equilibrium or yung equal distribution of uniform or equal distribution of molecules within a system or within a solution. So that's it. So it is the movement of molecules, no, or solute from higher concentration na area going to an area of lower concentration. So ito yung mga terms na kailangan nating malaman under the diffusion. You have the solution or a mixture. Ito ay tinatawag natin na homogeneous na mixture, no, yung solution. So any mixture of liquids, gases, or solids in which the substances are uniformly distributed with no clear boundary between the substances. So you cannot really distinguish the solute from solvent in a solution because they are uniformly distributed. Yan. So a solute dissolves in a solvent to form a solution. So yung solute, yun yung tinutunaw. Yung solvent, yun yung tumutunaw. And again, the primary goal of diffusion is equilibrium at kinakailangan merong concentration gradient or pagkakaiba-iba sa concentration. Okay? In different points of the system or the solution. So, dapat merong pagkakaiba-iba. Pag walang concentration gradient, guys, yung diffusion hindi makakapag-start. Okay? So, ibig sabihin, pag walang concentration gradient, baka nasa equilibrium na na-state yung solution. So, kailangan merong concentration gradient. So, in order for diffusion to start, dapat merong concentration gradient. So, yan. Okay? So, please take note of that. Yan. So, ano ba yung mga molecules na nagdi-diffuse inside and outside of the cell? You have there, number 1, lipid-soluble molecules diffuse directly through the plasma membrane. Kasi nga, lipids din itong ating plasma membrane. No? So, most of it is merong phospholipids. So, ito, lipids din. So, yung mga lipid-soluble molecules, madaling makakapag-travel or makaka-cross sa plasma membrane because of that nature of our plasma membrane. And yung mga non-lipid soluble molecules naman, hindi sila makaka-diffuse because of their nature. Nire-repel sila because here we have the lipid and then ito non-lipid soluble. So therefore, marirepel sila. But there are some specific non-lipid soluble molecules that can still pass through the plasma membrane through these membrane channels. So ito na yung mga nakilala ng receptor, for example, o di kaya may naka-attach na antibodies. So, pwede silang makapass through dito sa membrane channel or sa integral protein natin. So, again guys, for the lipid-soluble, pwede silang maka-cross directly without the need for the integral protein. Pero yung mga non-lipid, nare-repel talaga. No? So, hindi sila makapasok or hindi din sila makalabas. And of course, you have there the specific, no, non-lipid. Non-lipid sila pero makakapasok sila kasi nga recognize sila ng cell. So they will pass through this area dito sa integral proteins. So yan. So paano ba nangyayari yung diffusion, guys? So from an area of higher concentration, for example, in this setup, yung sugar cube, meron siyang higher concentration of the sugar, and then eventually it will spread, no, from area of higher concentration going to the area of lower concentration. So ganun hanggang maubos or ma-distribute equally yung or uniformly yung mga substances. So hanggang ma-achieve yung equilibrium. Pag na-achieve na yung equilibrium, titigil na yung diffusion. So yan yung guys. Yan yung for diffusion. So ngayon, papakita ko sa inyo itong video about diffusion.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient.
Okay. So, again guys, take note natin guys, no? Kung ano ba yung nagpapagalaw sa mga molecules. Bakit sila nagde-diffuse? So, please take note of the word kinetic energy. Yung mga molecules, guys, sa kahit solid na, no? Yung mga molecules, kahit solid sila, gumagalaw pa rin sila kasi merong amount of kinetic energy doon sa mga molecules na yun. And pag na-diffuse na sila, yung kinetic energy na yun, yung tumutulong sa kanila para mag-move from higher concentrated na area going to the area of lower concentration. So, ano yung mga factors na nakaka-apekto sa rate of diffusion? Again, dito guys, paki-take note dito. Yan. So, you have there, the rate of diffusion is affected by temperature, size of molecules, at saka yung steepness of concentration gradient or yung difference ng concentration gradient. Yan. So, paano ba nakaka-apekto yung temperature sa rate of diffusion or kung gaano kabilis yung diffusion? So, dito yung pwede nating gawin na example is yung pagtimpla ng kape. For example, yan. Yan yung pagtimpla ng kape. So, saan yung mas mabilis na diffusion or pagtimpla ng kape? Saan siyang temperature ng tubig mas madaling matunaw or ma-dissolve? Is it in the hot water or cold water? So, naturally, sa hot water siya. Kasi yung hot water, meron siyang kinetic energy. Yung molecules ng water dyan, nagmo-move na siya. So, pag nilagyan mo pa siya ng solute, mas gagalaw pa siya at mas madali yung diffusion of the solute. So, the higher the temperature, the faster is the rate of diffusion. Ano siya guys, no? Directly related. Pag tumaas yung isang factor, tumataas din yung isang factor. So, the higher the temperature, the higher is or the faster is the rate of diffusion. Pag bumaba naman yung temperature, bumababa din yung rate of diffusion or bumabagal din yung rate of diffusion. So that's it. Directly related sila. Again, the higher the temperature, the faster is the rate of diffusion. The lower is the temperature, no? The slower is the rate of diffusion. How about the size of the molecules? Yan. So ito naman guys is inversely related. So ibig sabihin pag tumaas yung isang factor, bumababa yung isang factor. Kasi nga, for example, pag mas malaki yung molecule, mas mahirap siyang i-diffuse. That's why sa ating body, most of the polymers or mga tinatawag natin na mga macromolecules ay bine-break down into simpler molecules. Kasi mas madaling i-diffuse, guys, yung mas maliliit na molecules kaysa doon sa mas malalaki. Just like, for example, yung starch, bine-breakdown pa natin siya into glucose para lang ma-absorb ng ating katawan. So, yung ano natin dito is, yung idea natin dito or yung conclusion dito is, pag mas malaki yung size ng molecule, mas slow yung rate of diffusion. Pag mas maliit yung size ng molecule, mas madali yung rate of diffusion. Okay? So, the smaller the molecule, the faster is the rate of diffusion. Yan. The bigger the molecules, the slower is the rate of diffusion. So, inversely proportional. So, paki-take note sa inyong mga notebooks. Okay? Again, for the temperature, directly proportional or directly related. The higher the temperature, the faster is the rate of diffusion. The slower the temperature or the lower the temperature, the slower is the rate of diffusion. In terms of size of molecules, the bigger the molecules, the slower is the rate of diffusion. The smaller the molecules, the faster is the rate of diffusion. How about steepness of concentration gradient? Yan. Ano ba yung ibig sabihin pag steepness of concentration gradient? Yan. Halimbawa lang guys, no? Halimbawa lang, meron tayo ditong dalawang container. So, eto, pareho sila ng volume. Container A, meron siyang dalawang sides. At dito naman sa container B, meron naman ditong dalawang sides. Merong membrane sa gitna. So, dito sa kabila, guys, yung concentration gradient ay, dito for example, is nasa 70%. Tapos, dito sa kabila ay nasa 30%. Yan. Tapos, dito naman, sa kabila, guys, is, for example, dito, 40%. Tapos, dito naman is 50%. Ano ba yung ibig sabihin ng steepness of concentration gradient? Yung difference ng concentration sa bawat side. For example, here, the difference is 40. No? 40% yung difference. Dito naman sa kabila is 10% yung difference. So saan kaya sa kanila ang merong mas mabilis yung pag-spread or pag-transfer ng molecules. Okay? Okay? So hindi natin pinag-usapan dito kung sino yung unang matapos na mag-diffuse, kundi kung sino yung mas mabilis na mag-diffuse ng molecules. Yan. So, sino kaya yung mas mabilis yung pag-transfer ng molecules? So, obviously, guys, the area or the container which has a steeper na difference or mas malaki yung difference niya. So, eto, guys. So, ibig sabihin, the steeper is the concentration gradient, the faster is the rate of diffusion. Okay? So, that's it. The steeper is the concentration gradient, the faster is the rate of diffusion. Isa pang context, no, or halimbawa, for example dito, meron tayong dalawang container ng tubig. Yan. Dalawang container ng tubig. Ang isa is puno. Puno ng tubig. Dito mayroong gripo. Pareho sila kalaki. Pareho rin yung gripo kalaki. At dito sa kabila, mga ganito lang siya. Kadami yung tubig niya. So, sino yung mas mabilis yung paglabas ng tubig, guys? Sino yung mas mabilis? Paglabas ng tubig. Yung velocity ng water dito, mas mabilis kasi mas malaki yung volume kaysa sa dito. Mas maliit, no? Mas konti yung tubig. Yes, madali siyang matapos, pero pero yung kanyang transfer ng water ay slower, no? So, yan. So that is how we illustrate the steepness of concentration gradient. Pag mas malaki yung difference sa concentration gradient, mas mabilis yung rate of diffusion. Hindi yung kung gaano katagal matapos yung diffusion. No. The transfer of molecules yung pinag-usapan, not the time that it would take to finish the diffusion. Okay? So the transfer, the rate of transfer, not the time na matapos siya. Okay? Kundi yung rate of transfer lang. Okay? Habang nagka-transfer yung molecules. So that's it. The steeper is the concentration gradient, the faster is the rate of diffusion. Again, for temperature, the higher the temperature, the faster is the rate of diffusion. For the size of molecules, the bigger the molecules, the slower is the rate of diffusion. So ano siya, no? Inversely related. Dito naman, guys, the steeper, pag mas malaki yung difference sa concentration gradient, mas mabilis yung rate of diffusion. Ayos ba? Okay, so paki-take note niyan, guys. Okay, so now let's go to the osmosis. Yan. So paano ba nangyayari yung osmosis? Ito naman ay isang klase ng diffusion, guys. So dito yung ating concern ay hindi yung hindi yung concentration gradient, kundi yung movement ng water. Hindi yung solute, kundi yung movement ng water ang ating concern dito. So, by the way, balik muna tayo dito sa diffusion. Ano ba yung summary ng description ng diffusion? Number 1, ano yung summary niya? Number 1, it is concentration-driven na klase ng transport, cell transport. Ibig sabihin, nakadepende sa concentration, guys. Okay? Concentration-driven. Number 2, it is a passive transport. So, ibig sabihin, hindi siya nangangailangan ng energy. Passive transport. And of course, number 3, yung movement is from higher concentration going to area of lower concentration. Again, concentration-driven siya. Number 1 ang description niya ng diffusion. Number 2, ano yung movement niya. So, number 2 dito sa diffusion is ano siya, no? Yung kanyang goal dito is equilibrium. No? And then, yung last is from higher concentration going to area of lower concentration. So, that's it, guys. Concentration-driven, passive transport siya. Hindi siya nangangailangan ng energy at from area of higher concentration going to area of lower concentration. And yung goal niya, guys, is equilibrium. Yan. Pag maabot na yung equilibrium, just like here sa illustration number 3 natin, pag naabot na yung equilibrium, magsa-stop na yung diffusion. So that's it. Okay? So paki-take note niyan. Napunta naman tayo sa isang klase ng diffusion which is called osmosis. So this is the diffusion of solvent, not the solute, across a selectively permeable membrane via diffusion. So through a specific channel protein we call aquaporins. Yan. So merong mga special na channel proteins na nagkikater sa transport ng water molecules. Yan. So tinatawag sila na aquaporins. Or pwede rin yung water na mag-pass through sa lipid bilayer directly. 'Di ba doon sa pinag-aralan natin sa parts of the cell or sa components ng plasma membrane, yung water ay pwede siyang makapass through. Kaso nga lang medyo hindi mabilis, no? Mabagal siya. Kasi nga sa polarity nila, no? Yan. So that's it. So we have here the different terminologies naman. You have the osmotic pressure or the force required to prevent the movement of water across a selectively permeable membrane. And then isosmotic solution, ito yung mga solutions na merong same ang concentration yung solute at saka yung solvent. Sa hyperosmotic naman guys, greater yung concentration ng solute kaysa sa solvent. Yan. And then sa hypoosmotic naman, lesser yung concentration ng solute kaysa sa kanyang solvent. Yan. So dito for example, sa hyperosmotic na solution, pag nag-mix ka ng tubig at saka water, masyadong maalat yung water. Dito naman medyo malabnaw. Yan. Malabnaw yung lasa ng water. Ibig sabihin, mas madaming water dito kaysa sa solute. Dito naman, mas konti yung water. So, medyo concentrated yung solution na ito sa hyperosmotic. Ito yung mga nangyayari guys sa ating dugo. Yung condition ng ating dugo. Every time na merong abnormalities na nangyayari sa levels ng blood sugar, for example, or sodium or salt sa ating blood, nakaka-apekto siya sa functioning ng ating mga organs at siyempre cells, no? So yung osmosis guys, yung movement ng water ay hindi lang siya nakadepende sa concentration gradient, kundi nakadepende siya sa osmotic pressure. So instead of concentration gradient, here in osmosis, water molecules move from area of higher osmotic pressure going to area of lower osmotic pressure. Kuha ba? So naka-pressure siya. Hindi siya naka-concentration gradient. Of course, yung concentration nagdudulot ng pressure. Pero dito, tignan na lang natin siya sa pressure na perspective. So pressure yung ipa-prioritize natin. Okay? So from area of higher pressure, osmotic pressure, going to an area of lower osmotic pressure. So tulad nito guys, this one is a selectively permeable na membrane. Etong flask or yung tubing natin, meron siyang selectively permeable na membrane. At dito sa itaas, meron tayong 3% ng salt solution. So merong salt dyan, merong water, sodium, chlorine, meron dyan. Sinubmerged siya ngayon sa distilled water, no? Sa beaker ng distilled water. So yung nangyari guys is, since marami ditong water kaysa dito sa loob ng ating tubing, yung water dito or yung osmotic pressure dito is mas malaki kaysa dito sa may osmotic pressure sa loob ng tubing. So ano ang mangyayari? Yung water molecules will move from area of higher osmotic pressure papunta doon sa area of lower osmotic pressure. So yan, tumaas yung level ng tubig dito. Tumaas yung level ng salt solution because of the entry of water molecules. So bakit hindi lumabas yung salt solution? Bakit walang sodium o chlorine na lumabas dito? Dapat lalabas din sila, di ba? Kasi nga, hindi sila makalabas, guys. Masyadong maliit yung pores or yung daanan ng molecules dito sa selectively permeable. Tinawag ngang selectively permeable, guys. Kasi nga, hindi lahat pwede makapasok or pwede makalabas. Only water molecules dito. So, nandito lang yung salt solution at pinasukan pa ng maraming water kaya nag-increase yung ano guys, no? Nag-increase yung level ng tubig. And for diffusion, di ba yung goal ng diffusion ay equilibrium? So dito sa special case ng diffusion guys, which is osmosis, yung goal nito ay ma-achieve yung tinatawag natin na hydrostatic pressure. O yung pressure ay equal na both inside and outside of the tubing, or in the context of cells, syempre, inside and outside of the cell. Yan. Pag ma-achieve na yung equal na osmotic pressure sa both sides, tawag natin dyan is hydrostatic. Pag static, di ba, wala nang galawan. Static, no? Stationary. So, dito guys, hydrostatic pressure yung goal ng ating osmosis. For diffusion, that is equilibrium. So, general term yung equilibrium. Dito naman, hydrostatic pressure. Kuha ba? Yan. Kung bakit hindi lumabas yung salt solution, that is because selectively permeable siya. So, dito, papakita natin kung paano ba mag-move yung water molecules across a selectively permeable na membrane at sa impermeable na membrane. So dito, we have the membrane na permeable to solute. Ibig sabihin, water molecules at saka yung solute pwedeng makakross. Dito naman, impermeable to solute. Ito na yung selectively permeable. Ibig sabihin, only water molecules can move across the membrane. So, eto guys, meron tayong dalawang klase ng osmosis dito. Osmosis sa membrane na permeable also to solute at osmosis sa membrane na impermeable to solute. So, unahin natin yung permeable to solute. So, ditong setup guys, ibig sabihin dito, yung ating membrane ay ina-allow niya both water molecules at saka yung solute na gumalaw across. Okay? So, umpisa natin. Saan kaya gagalaw yung molecules ng solute at saan kaya gagalaw yung molecules ng ating water. So, again, mapapansin ninyo guys na mas maliit yung bilang ng solute molecules dito kaysa dito sa kabila. Ibig sabihin, hindi sila equal ng concentration. So, ang tendency pag solute yung gumalaw, dito siya manggagaling, no? From area of higher concentration going to area of lower concentration. So, dito 4 lang. Dito sa kabila, 1, 2, 3, 4, 5, 6, 7, 8, 8. So, ilan kayang molecules ng solute yung magta-transfer para ma-achieve yung equilibrium dito? Ilan? So, 2. Okay? So, 2 siya. O, tingnan nga natin. Yan. Gumagalaw na yung molecules. So, ilan yung nag-transfer? Ayan. Okay? So, 2. 2 yung nag-transfer. Kung makikita nyo kanina, yan. Ulitin natin. 2 yung nag-transfer para magkaroon ng same amount or same na bilang yung solute. So, 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Pareho ng 6, equal na yung concentration ng solute. How about water? Kasi yung ating ditong concern is water, osmosis ito eh. So kung makikita nyo guys, hindi lang solute yung gumalaw, kundi pati na rin yung water molecule, and that is to achieve what we call hydrostatic pressure. So kung ang solute, yung kanyang goal dito ay equilibrium, ang sa water molecule naman na paggalaw, ang kanyang goal is hydrostatic pressure. Para mayroong parehong osmotic pressure yung both sides ng container. Kuha? So, ito guys, yung movement niya. Okay? Again, yung ating, membrane ay nag-aalaw both sa solute at sa water molecules na mag-cross. Yan. Ngayon, punta naman tayo sa selectively permeable na membrane. Ito, hindi na makagalaw or hindi na makalabas dito or maka-cross yung solute. Yan. Hindi na siya maka-cross dito sa susunod natin na setup. Eto guys. So yung membrane natin is only permeable to water. Ibig sabihin hindi pass through yung solute neto. Yan. Kahit 4 lang sila or kahit dito maraming solute, hindi sila maka-cross dito sa membrane natin. Only water molecules. So ano kanyang mangyayari guys? So dito guys, makikita natin yung pressure na, osmotic pressure. So since dito yung ratio ng water sa may solute molecule ay mas malaki, ibig sabihin yung osmotic pressure dito ay mas malaki rin. Okay? While dito, mas maraming solute at marami ding water molecules, pero yung kanilang yung kanilang ratio ay mas maliit kasi mas maraming solute dito. So ang tendency is water will move from area of higher osmotic pressure going to an area of lower osmotic pressure hanggang maabot yung tinatawag natin na hydrostatic pressure dito. So bakit kaya guys na yung movement is pa ganito? Yan. Bakit kaya pa ganito guys? So again, kanina yung basis natin is yung ratio. Kasi nga guys, yung interaction dito kanina, for example here, yung interaction kanina ng ibang water molecules sa may solute molecules ay nag-iiwan ng mas maliit na bilang ng mga free water molecules. So only few will be left na free. Kasi yung ibang water molecules mag-interact sila with the solute molecules. So bababa yung osmotic pressure dito. Kaya yung tendency is water molecules will move from area of higher concentration or higher osmotic pressure going to an area of lower osmotic pressure hanggang ma-achieve yung equal na ratio sa both sides, no? Ratio yung pinag-usapan natin dito, hindi na siya concentration. At saka yung pressure. Kasi yung ratio nagdudulot siya ng pressure dito. So from area of higher pressure, higher osmotic pressure going to an area of lower osmotic pressure. So yan guys. Okay? So tandaan natin kung paano ba gumagalaw yung mga molecules under osmosis. Okay?
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
Okay? So dito guys, makikita natin na yung interaction ng water molecules, although na mas marami sila, yan. For example, magkapantay, no? Magkapantay lang yung amount o yung bilang ng water molecules sa magkabilang side, ay yung nagpapaliit dito guys sa amount of free water molecules ay yung solute. Kasi yung ibang water molecules na available, for example, 20 dito na water molecule, tapos dito 20 rin. Kaso nga lang meron tayong solute dito. So yung ibang bilang o yung ibang water molecules will attach to the solute. Mag-i-interact sila. For example, merong 6 na nag-interact with the solute molecule. So, ilan na lang yung matitira dito? Merong 6. So, merong 14 na lang. No? 14 dito, 20 pa rin dito. 20 kasi wala naman sila nag-interact. Hindi naman sila nag-interact sa isang solute. So, 14 na lang dito, 20 na lang dito. Or 20 pa rin dito. So, yung osmotic pressure dito, mas malaki kaysa dito. Kasi mas maliit na lang yung bilang ng free water molecules. So, ang tendency is, the water molecules will move from area of higher osmotic pressure going to an area of lower osmotic pressure para ma-achieve yung tatawag natin na hydrostatic pressure. Yan. So, ganun yung nagiging resulta ng osmosis. Okay? So that's it. Okay. So now let's go to the reaction of the cells sa iba't ibang solution sa osmosis. No? Osmotic concentration of solutions. So meron tayo dito, for example, sa animal cell, we have here, and for the plant cell, also, yan. So, pag-usapan natin is yung kanilang magiging reaction. For example, in a hypotonic na solution, for the animal cell, ano yung mangyayari? Katulad neto, you have the RBC. Ano yung nangyari? Nag-burst siya. Yan. So, kasi, sa hypotonic na solution, yung water, yung osmotic pressure sa labas ay mas mataas kaysa sa osmotic pressure sa loob. So, ang tendency is mag-move in yung water molecules hanggang sa pumutok na lang yung cell ng animal cell, no? Yung animal cell. Yan. For the plant naman, ano kaya yung mangyayari, guys? Puputok ba siya or hindi? So, ano yung mangyayari? So, magsiswell lang siya. Mamamaga lang. Magsiswell lang siya, guys. So, hindi siya magbo-burst. Magsiswell lang siya. Bakit kaya magsiswell lang siya? So, bakit magsiswell lang yung plant cell? Yan. Bakit kaya? Kasi nga, meron siyang cell wall, yung plant cell. How about yung bacterial cell? O yung mga prokaryotic cell? Bacterial cell, ano yung mangyayari? Ganon din, guys. Ganon din, guys, no? Magsiswell din sila. Kasi nga, meron ding cell wall yung bacterial cell. Unless yung bacteria ay walang cell wall, no? Pero, karamihan sa mga bacteria, merong cell wall, eh. Yan. So, magsiswell din. How about for the isotonic solution? So, sa isotonic solution, guys, pareho lang yung net movement ng solute at saka ng solvent or ng water at saka ng solute in and outside of the cell. So, walang mangyayari. So, no change, no? No change. Walang changes dyan, no? No change sa cell. So, sa animal cell, sa plant cell, sa bacterial cell, walang mangyayari kasi nasa isotonic solution. So, ibig sabihin, yung osmotic pressure in both sides, inside and outside of the cell, ay pareho lang. So, yung net movement, pareho din. So, walang mangyayaring change dyan. Okay? So, that's it. And then, what else? You have here, the last one, the hypertonic solution. So, anong mangyayari sa hypertonic solution? Anong mangyayari sa animal cell? Anong mangyayari sa plant cell? At anong mangyayari sa bacterial cell? Sa bacteria. Yan. For the animal cell, eto, obvious na, napakita na. So, magsishrink siya. Yan. Or, yan, no? Magsishrink siya. Magsishrink. Kasi nga, mas malaki yung osmotic pressure sa loob ng cell kaysa sa labas. So, ang tendency is, lalabas yung water. So, magsishrink. No? Magsishrink yung cell. Yan. Crenation yung tawag dyan. No? So, magsishrink siya. How about sa plant naman, guys? So, yung plant, hindi siya automatically magsishrink kasi nga meron siyang cell wall. So, ang mangyayari sa may plant is yung kanyang plasma membrane or yung kanyang cytoplasm lang yung magsishrink. Yung kanyang cytoplasm yung magsishrink at may iiwan yung structure ng kanyang cell wall. So, ang tawag dyan, guys, is plasmolysis or magpa-plasmolyze siya. Plasmolysis. Magpa-plasmolyze siya. Yan. So, yung kanyang, yung kanyang cytoplasm lang yung magsishrink, hindi yung buong structure ng cell. And then, for bacterial cell, malamang ganun din. No? Magpa-plasmolyze din kasi may cell wall din sila. Yan. Plasmolyzed. So, that's it. Kikrenation siya. So, that's it for the osmotic pressure. So, ito yung summary. No? For hypotonic solution, magbo-burst yung animal cell kanina, yung plant cell magsiswell, yung bacterial cell magsiswell din. For isotonic, no change. For hypertonic, magsishrink yung animal cell, yung plant cell naman magpo-plasmolyze, bacterial cell will also plasmolyze. So that's it guys for the osmosis. Okay, so kung may mga tanong kayo regarding osmosis, paki-message lang sa messenger or di kaya sa email. So maraming salamat.